This tutorial is going to take a look at this practice problem from Lecture 2.5. If you haven't had a chance to look it over recently, please pause the video now and take a minute to read the question, then come back to see how we're going to tackle it. The first thing that we need to review before we try to answer this question are transposons. Transposons are mobile genetic elements, or jumping genes, that move around the genome. Let's take a look at one way that they can interfere with gene function. Here we have a gene of interest. The transcribed region of this gene is highlighted in orange. When this gene is expressed, it is transcribed and translated into the normal or wild-type protein, and this protein will be able to do whatever it is that it's meant to do. So now let's say that a transposon comes in and decides to insert itself into this gene as shown here. If a transposon inserts itself into a gene, there is a very good chance that it will disrupt the function of that gene. Genes can't produce the right protein if they're interrupted by an unexpected transposon sequence. In this case, the transposon has inserted itself into the transcribed region of the gene. And now, if this gene is transcribed with a transposon a part of it, it will be translated, if it's translated at all, into a mutant protein that probably can't function normally, resulting in a loss of function mutation. Some transposons, like this one, preferentially insert themselves into transcribed regions of genes. These can disrupt gene function by disrupting transcription, RNA processing, or the protein sequence. Other transposons are more likely to insert into regulatory sequences or the promoters of genes, in which case they will most likely affect transcription. One of the things that makes transposon insertions interesting is that they produce one of the few kinds of mutations that can be detected by PCR. Let's say you have a set of primers that can be used to amplify or make many copies of this gene. The forward primer binds just upstream of the transcribed region, and the reverse primer binds just downstream. If you do a PCR using these primers, you'll produce a product that includes the transcribed region of the gene of interest, plus a bit of extra sequence, up to and including the primers. If you use this same set of primers to amplify the version of this gene that has a transposon insertion, the mutant PCR product will include the transcribed region of the gene, the transposon, and a little bit of extra sequence, again up to and including the primers. Since the mutant product includes the transposon, when you compare the lengths of the two PCR products, you can see that the mutant PCR product will be longer than the wild-type product. The difference in size between the two products can easily be detected by gel electrophoresis, and we can go back to the question now to see what this means in terms of analyzing our data. In this question, we know that the transposon insertion interrupts a gene required for pigment production in corn kernels to generate a loss of function mutation. So this means that the wild-type version, which does not contain the transposon and so is shorter, will be the functional variant. This variant allows for pigment production, so kernels with this variant will be pigmented. The mutant version, which contains the transposon and so is longer, is a loss of function variant that cannot produce pigment. So if a kernel contains only this variant, it'll be white. We see this in kernels 1 and 4, which possess only the transposon-containing variant. In kernels 2 and 3, they possess only the wild-type variant. Since the wild-type variant produces colored kernels, kernels 2 and 3 will be colored. Since the mutant variant produces white kernels, kernels 1 and 4 will be white. Then the question becomes, what do you expect from kernel 5? For this, you'll want to remember that often, a single copy of the wild-type variant is sufficient for function. Since kernel 5 has one of each version of the gene, it should be colored, since the wild-type copy of the gene will allow pigment production to occur. And while our question focused on the basic question of pigmented versus non-pigmented kernels, I would be remiss if I didn't acknowledge that within the category of pigmented, there can be lots of variation. Have you ever tried corn that looks like this? It's known as glass gem corn, and you can grow it yourself. There are almost certainly a number of different genes that are responsible for the variety of colors that can be produced by glass gem corn. The details aren't worked out just yet, but that just means that there's still an opportunity to learn more about it.